Great, thanks, Antonio, and thank you again, everyone attending today. So what we're going to do today is we're going to talk about Ingenuity Variant Analysis, and what I've done today is put together a few items that I think will help all of you work with the application more effectively. Just some objectives of what we're going to be doing today is really to show you ways you can work more effectively and make better use of your time using the application. I know I work with people all the time out in the field who are using variant analysis, and I kind of see where people get stuck and some of the things that cost them time, where there's some easy solutions to help them out. The other objective is really to discuss ways to make your sequence variant analysis more robust and accurate. So we're going to spend a bit of time today talking about how to use the filters, how not to over-filter by accident. There's some things that people I see doing out in the field again that I instruct them on to say, well, you probably don't want to do that because of X, Y, and Z. So we're going to cover a few of those today. And then lastly, one of my assumptions is that everybody who's on the phone today is at least relatively familiar with Ingenuity Variant Analysis. Meaning you've seen the application, you're familiar with the display and the filter cascade and the general concepts of how it works. So I'm not going to spend a lot of time, talking about the basics. We have lots of other videos that will do that. Today is really working on the assumption that you are familiar with the application. So the first thing we're going to do today is walk through the agenda. So we're going to talk primarily on how to get started more effectively. So analysis setup tips, how to quickly upload and analyze your variant analysis. Then we're going to talk about artifacts, and this is one of those places where a lot of people get hung up. They either assume too much or too little around how much false positive and false negative data there is in a sequencing analysis. So we'll talk about the use of controls, we'll talk about how to remove some of those artifacts, and then we'll talk about specifically some of the filter settings. We'll hit primarily on the predicted deleterious filter and the genetic analysis filter, and then just kind of how to, and you'll see as we go through today, just how to be creative with some of these filters in the filter cascade and analyzing those variants. When we get more toward the end of the analysis or end of the presentation today, we'll be assessing variants. So I have kind of a handful of items that I usually will walk through when assessing a variant to see, is it artifact? How biologically compelling is it? And then lastly, a lot of people ask me, well, how do I save what I've done? Or if I want to do a forked analysis and ask separate questions, which is one of the things I actually encourage, how do I save each one of those off and have different versions? So we're going to talk about that. Another common one is how do I save my filter settings and reuse those filter settings, and so we'll talk about that as well. There's some definite tips and tricks there. So let's begin by marching into the analysis setup tips. There's a few things here on my slide here. So one is to use the high volume data uploader, and I'll go through each one of these. I'll just show you briefly. How to annotate samples for quick sample lookup. So we can talk about how to annotate, how to give attributes or clinical features to some of your samples for lookup, how to add subject IDs, and then we'll talk about just kind of the way I like to set up my analyses. A lot of these are more for people who have more samples as opposed to fewer samples, but I think there's probably some tips here that'll help. So what I'm going to do now is I'm going to hop on over to the application and just touch on a few of these things that were in my slides. So the first one there was the high volume data uploader. So the first step into using variant analysis is obviously you need to get your VCF, your variant call file, into the application, and there's an upload button up here. So my assumption is that you all know how to get here and you all know where your samples table is. So I'm going to click the upload button, and a lot of people know how to enter their VCF data or point this to a file of VCF or complete genomics file. But there's a link down here that I just want to point out to everybody because it's easily missed. It says use our high volume data stream uploader. And honestly, I use this data uploader if I have more than anything like 5 files. I'll go ahead and use this way of uploading my data rather than the onesie-twosie way up here. If I click on that, you'll see a browser window comes up, and then I can go choose my file. So I can actually go point to a directory and select all my VCF files from a single directory, click upload, and essentially not worry about it for a while. One of the things I've noticed is about half the time this window does not appear because pop-ups are blocked. So if you want to use that high volume data uploader, just make sure your pop-ups are enabled and you're allowing this this additional browser window to pop up. I'm also going to point out, this is more for the more daring, we do have a download manifest template that you can use. So if you want to upload some of the annotations that I'm about to talk about, if you want to do that at the time you set up your analysis, you can do that and include this with your variant files. So that's one way to kind of get started a little bit faster, get things into the system a little easier. I will also just mention in passing that we also have an FTP pipeline that we can set up for you too. So if you are, really, if you have a lot of samples and you want to be able to in an automated way send them to variant analysis, we can set that up for you as well. So once in the application, let's say you've used a high volume data uploader and you have a bunch of samples. So here's an example where I have a whole bunch of PGP samples. This is a personal genomics project or a personal genome project. And so I have a bunch of those samples here loaded into my system. And down below this I have one of our published papers data from the NCI 60 cell line. So I have this data down here. And you can see there's a lot of samples here. And so one of the issues is, well, how can I find some of this data? How can I better utilize it? And one of the ways is to actually give an annotation to it. So you can see when you upload data, it'll sometimes come along without very much information. Just one field gets reproduced multiple times in all my fields. And now my goal is to come back and edit these. Now one option to edit is I can select each one, and there is an edit button that I can click. And I can come in and I can change the display name, the subject ID, and the description if I so desire. Obviously, you can imagine with all of these PGP samples that would be pretty tedious. So there are some other ways to go about annotating samples, and another way is to use the annotate button. This is pretty well described out in our help, so you can go to our help and learn more. But what I can do is essentially add what we call clinical features, or I just call them annotations or attributes about these samples. This is primarily for lookup. This is not for statistical testing or anything of that sort. But I can point this to a file, and I'll just bring up an example file. So here's an example file, and in this case I'm using subject ID as my key. I could also use the barcode or display name, so it's kind of up to you what you want to use as your lookup. But so I have these three samples that I've already uploaded. If I want to annotate them with any sort of value, and I can just invent what value I want and put it as a column, and I can then give the, we would call these keys, I can give them values down below, and I can do that for any or all of my samples. I would then point this to that file. That was in Excel. I'd save it as a tab delimited file. And then I would have those annotations attached to samples. So you notice I annotated several of my samples with neuroendocrine carcinoma. So if I want to come in and type neuro, and I don't even have to get that far, you can see I have some hereditary pheochromocytoma samples here in my sample directory just by searching for them. Over on the right hand side, if I select one, you'll see it says tumor, neuroendocrine carcinoma. So tumor and neuroendocrine carcinoma is what I had in that file as my annotation. So now I can do a quick lookup for all neuroendocrine carcinomas in my data set. So it's another way of annotating samples for a quick lookup. Okay, so that's another speedy way to get started and subset samples for your analyses. I'm going to go ahead and clear that out, and let me give you one more tip here. This is just something that I like to do. So let's pretend I want to use these PGP samples and I want to use these NCI samples in analysis, and there's a lot of them. What I like to do is select my cases first. So you'll notice I have all of my NCI samples say NCI in them. So I'm going to come up here and I'm going to type in NCI, and what that's going to do now is it's going to limit my samples here to just the NCIs, and I have a couple of others that have a string match that I'm going to ignore down here at the bottom. I'm going to go ahead and select the bottom one. I'm going to select the top one now. Now all my cases, what I'm going to use as my cases, not my controls. My PGPs are going to be my controls in this analysis. I can go ahead and now click Analyze, give this a name, pretend that this is going to be a cancer analysis, and again, hopefully you all know this is not make or break, right? The whole idea is that this is going to help set up the filter cascade in your analysis, so don't get hung up on, gee, what button do I push? I know that a lot of times studies will kind of straddle a couple of these radio buttons here. Just pick one. We'll get into the filters and then make the adjustments as necessary. Click Next. Let's go with any cancer. Now, by selecting all of my tumors first, or all my cases first, it's going to put them into my case window over here, so you can see all those NCI-60 cell line samples are all up here in my cases. And now I can come here in this search bar and type PGP, and again do something very similar with my PGB samples, and I can use these now as controls. So you can see how, particularly with large samples, it's really nice to use these lookups to then select my samples and put them together. I'm not actually going to analyze this. I already done that. So those are some easy wins. Let's go back to my presentation here. Of using the high volume data set uploader, adding annotations. I didn't mention it, but you can also add subject IDs, which is really important. I guess I'll go ahead and bounce back real quick. When you're in the samples here, if you have tumor normals that you want to pair up, so you can do essentially a somatic subtraction, the subject IDs, which essentially you can think of it as a patient ID, needs to be identical. So if you focus on these samples down here at the bottom, you can see I have 2 cell lines, HCC 2218. You can see that I have 2 of them. One of these is blood, which would represent germline in this case. The other one is a ductal cell carcinoma. And so that is the other one here. They have identical, no white space, nothing different between these 2. So when I send these 2 to an analysis, it'll allow me to do that tumor normal pairing in my genetic analysis filter. I'll touch on that again later as well. So that covers some quick tips and tricks on how to upload and analyze your samples quickly. The next section that I wanted to cover was artifacts. So this is has been since the beginning of variant analysis an issue for our customers. You know, it's nothing having to do with variant analysis; it's everything to do with the assay of sequencing. And you know, one of the things I tell everybody when I'm out in the field, I say, well, you know, sequencing and processing artifacts, they are your enemy. They are the thing you have to deal with the most. You have to keep in mind that sequencing, like any scientific assay, is subject to false positives and false negatives. And so, like any good experiment, it's always good to have controls. Artifacts can be introduced at several stages. You know, there's the library prep, there's PCR if you're not doing PCR free, there's the sequencing aspects of it, there's the aligning aspects of the sequences, there's the calling. There's just so many places. In all of those places, we can get artifacts. Some of those artifacts are reproducible, like MUC4. I see all the time. Doesn't matter what platform you're using, doesn't matter how you're aligning those. MUC4 is like in every single analysis I see. Some of them are platform dependent, so it makes a difference. Are you using complete genomics? Are you using Illumina? Are you using GATK for your aligning? Are you using, you know, something else? So some of them are going to be very specific for your platform, but for that platform they are relatively reproducible. On top of that, there are sporadic artifacts, meaning there's just randomness. Think about, you know, there's just entropy in the system, and so there's just going to be random false positives, essentially, that are going to be in the sequencing data. So you just have to be aware of that. It is your enemy, and you need to guard against that enemy at all times. One of the ways you can do that is to use controls when you can. Okay. The best controls, like, again, kind of just going back to basic scientific technique here, the best controls are going to be normals that are prepared, they're sequenced, they're aligned, they're called, they're analyzed together, they're taken through the whole pipeline side by side. Okay. Now, a lot of people don't do that, and also in some cases I think it becomes prohibitively expensive. So another way to go is, well, you know, using the same platform, aligner, caller, etc., but maybe just not side by side, maybe done at a different time. That's another really good control you could use. Maybe your buddy down the, you know, who's also using variant analysis has some controls and you'd like him to share them with you. Maybe they were done on a different platform, or you don't really know how they were used. It's still probably better than nothing. But that being said, it's not going to be quite as robust at getting rid of some of that noise that's platform specific. And I kind of put this bullet here. When analyzing cancer samples, obviously you'd like your normal tissue to represent your germline sequence. They need to be paired up with your tumor, and that way you can identify variations that's specific to your tumor. Okay, so in that case, it's really important to have a tumor normal control, or at least it's helpful. Let's put it that way. If you don't have controls, don't worry about it. You can still do your work. It's just you're going to be a little bit more susceptible to that artifact. Now, in talking about controls, I mentioned that some are platform dependent, and that's very true. So especially if you're taking controls from somewhere else, it's kind of nice to know kind of how well they mesh up with your platform. And one way to do that is to look at the summary within variant analysis. So you can look at the distribution and makeup of variants at the predicted deleterious filter and see how they compare. You know, good patient matches, if they're done on different platforms, may not necessarily be good controls. Although, again, some controls are better than no controls. So here's a screenshot of the application. I'm not going to open this up. If you remember that PGP NCI 60 analysis that I started out with, well, this is it. This is a screenshot of that analysis. And you can see in the application, I'm over here on the predicted deleterious filter, and I started out by clicking over here on the variance, and then I came up here and clicked on the summary, and then I went from there, said, well, I'm interested in variant type, and then from there, and this is a button my colleague made me aware of actually. If you click here, so by default you get the pie chart, but if you click, there'll be a bar chart symbol there. If you click on that, it'll actually show a bar chart where each bar here is actually one of my samples. So the way this breaks up is, let's see if I can draw kind of a straight line through here. The samples over on the left are my NCI samples, and the samples over here, which I'm using as controls, are my PGP. And what you can see here is that the blue actually represents the substitutions, okay? And then we have, I believe this is deletions and insertions, top and bottom. So you can see that there's a pretty different profile between my controls versus my cases, and that's because these are from two completely different sources. Actually, one's exome, one's kind of a modified whole genome. So they're very different in their composition. That's not to say that they're bad controls. Again, some controls are better than no controls, but it just gives you a sense for, you know, kind of some differences that you should be aware of when looking at your data. So here we are looking at another analysis. I just want to highlight the artifactual aspect of sequence data. This is looking at 20 samples, and here I've gone through and I've done a quality score 20 and common variant, so I've removed anything greater than 3%. And then kind of my standard predicted deleterious. This is again a screenshot. We'll go live here in a minute. And here's my genetic analysis filter where I'm looking at pretty much any genotype, because these are just cases. I don't have any controls in this particular experiment. And I end up with all of these variants at a very high frequency within the set. And you can just look at this and you can say, this is interesting. You know, how do you tell what's good from what's bad? This is all pretty much artifact. These are my PGP samples. These are my controls. And you can see it's just littered with what looks to me artifacts. Because I've already filtered against 1,000 genomes, complete genomics genomes in the exome sequencing project. So there's nothing here that's indicated to be in the population more than 3%. But you can obviously, in my study, it's 40, 50, 80%, whatever. Let me jump away from this. Let's go back. Let me jump back to my analysis here. So here's a slightly different version of this. So now I'm live back in the application. This is actually 60, so this is a much larger number of my PGP quote-unquote controls. And so now, because I have a much larger number, each variant gets represented as just a line instead of a bar chart like we were just looking at. I wanted to show you the bar chart just to give you a sense of some of the heterogeneity and genotypes and whatnot. So, as I mentioned, we have confidence at 20% quality score, common variance. I'm filtering out anything more than 3%. Predicted deleterious. I got pathogenic, some gain of functions, and pretty much coding or splice site loss of function in my predicted deleterious filter. And here on my genetics filter, what I've done is I've actually filtered for cases. Well, 38. I was actually shooting for 40. I can go ahead and rerun this. Or how often do I see a variant occur in 40% of my cases here? And you can see it is still huge. I still have 150 variants, and some of these are disguised as looking very interesting. For example, if I look at this, OR2T12, I have a variant finding here, and if I go look at that variant finding, this publication around it, something that was published in PLOS One back in 2013, last year, had a somatic missense heterozygote of this gene is associated with astrocytoma, and there's a cosmic finding as well. But you can't tell me that that this is so critical in this particular sample. Here's another one where if I go look at the variant finding, I have 3 cosmic entries here, all different study sizes, and this is information that we've curated from our knowledge base. By the way, I'm not the cosmic. We pulled in the cosmic, but like that, the PLOS article, we're investing a lot of money in curating this variant content from the literature. But you can see people are reporting these variants as being important. You can see the frequencies down here are all essentially 0. So this is all pretty key artifact. Now we have some tools to get rid of this. One of those tools you can find in the confidence filter. So we're going to show you how do I get rid of this nasty noise, this artifact. Well, we have these noise filters down here, and I'm actually going to put this filter again. So this is something that you can do. So I'm going to go ahead and close this up. I'm not going to implement it down here. I'm going to add another filter. I'm going to add another confidence filter down here at the bottom, and I'm going to add our noise filter. So we have a noise filter that will remove anything outside the top X%. Here we have 0.2% most exonically variable 100 base pair windows in healthy public genomes. I have a slide that we'll discuss this in a minute, but essentially it means we're going to slide across the genome in 100 base pair window, and we're going to find out which variants are the most variable across the 1,000 genomes. And then down below that, we're going to do it at the gene level. Okay, and I can apply that here, and you'll see that I go from 150 variants down to 84. So it does a pretty good job at the default settings getting rid of that data. And just to give you a sense, again, for what that first tool is doing, the one is doing it on a sliding window. So pretend we have 1,000 genomes all lined up down here, and we take our 100 base window and we slide that along the genome, and then we count in each frame how many variants are now in that 100 base window. And then we basically say, well, given the percentile, let's remove or ignore X%. And the wording is a little funny. We say keep outside the top percent. So the outside would be the stuff in the red box, and we're going to get rid of anything that looks to be extremely variable. So that's what that first checkbox does. That second checkbox does it at a gene level. This is something I did a long time ago. This is looking at a single sample where I just counted up variants per gene. Here's our favorite muck. You can see there's a whole bunch of genes that just have a lot of variants in the genes. But if we look across the entire analysis, going to this next table, you can see, well, how many genes have one variant? Well, almost 4,000, which actually accounts for 56% of my genes in my analysis. And if we just go up a little ways, you can see that if I look at this one sample, 90% of my genes have 4 or fewer variants. Okay, but if we keep walking across this table, you can see that 4 or fewer variants, so 90% of my genes only make up about 60% of my variant content. So that means there's a bunch of noisy genes from here on out, right, that are making up, roughly 40% of my content is coming from 10% of my genes. Okay, and you can draw the line wherever you want, and that's essentially what we're doing when we are back here in the application and we're setting that confidence level. I'm actually going to add another confidence filter here just for fun, and I'm going to crank these babies up to 5. This is actually what I normally do. This is my tip here. This is my acid filter. If you can get past this filter, you're probably not too noisy as far as the 1,000 genomes go. So here we're going from 150 down to 84 down to 63. We could even get more stringent than that if we wanted to. We could even, take this out to 10%, which is probably not a terrible place to be. Now, you're going to be throwing some gene content away, but, when you have good variants hiding out amongst a bunch of artifacts, it's questionable whether you're going to be able to make sense of that. So you can see we're down to 51, so we really cranked this down. There's still a whole bunch of variation over here that's still pretty noisy, and you're probably wondering, okay, well, great, now what do I do? Here's a solution you can consider. So you can take an analysis like this and run the analysis, and then come up to your genetic analysis filter and decide, at what level, because these are going to be very platform specific, remember, right? Because we are basing our analysis off 1,000 genomes. So this is noise that's not appearing in the 1,000 genomes data, but it's appearing in the PGP data. So I can decide, well, let's say, I want to filter out anything that occurs greater than 10% in my controls, and I just want to use that going forward. So I'm going to go ahead and apply that. Hopefully this won't take very long. There we go. So I've applied that now. I'm going to come up here to my genetic analysis filter, and you can see that I have 1,400 variants, 1,439. And now if I want to use this and say I want to use this as the filter for subsequent analyses, okay, I can create a list, and I can call this list PGP, let's call it PGP 20. Okay, so anything greater, or sorry, that was a 10% cutoff I did, PGP 10. So anything greater than 10 or greater, it's going to be in my list. One caveat, once you make a list, right now we don't have a way to delete it, so make your lists sparingly. So now I have a list that I've saved, and now I have the ability, and I'm just going to show you in this one, to then add another filter called user-defined variance. And now I can select that PGP 10 list and apply it. Oops, and actually I missed one step there. I want this to be an exclude. I don't want to keep my artifacts, I want to exclude them. Okay, so now it's an exclude filter, and now I can move this up in my pipeline. And it doesn't matter where I moved it, obviously anywhere below the genetic filter. It's going to get rid of my variance now. So the nice thing is, though, that list can be accessed from any analysis. So if I'm doing another analysis on another set of samples that are my case samples, my disease samples, I can add my user-defined variant filter, and I can pull that PGP 10 list, and I can use it in that other analysis. So it'll always be accessible to me, and I don't have to load up my entire bank of PGP samples as a control every time. Okay. The one caveat to that is that we are removing at the variant level, so we're not removing at a genotype level. But essentially, what I've done is I've created a filter that says if that variant occurs at greater than 10%, I can use it. So I'm essentially creating my own control filter here. So hopefully that was helpful. That's I think one of the key things a lot of people struggle with is really getting through some of that artifact data. So just to review, obviously artifacts are your enemy. Use controls when you can. The best ones are paired ones, but any control is better than no control. Assess controls by analyzing and comparing in that summary view. Remember to hit the bar chart symbol. Use the genome variability settings in the confidence filter to improve probable false positives, or I should say remove probable false positives. And that's based on 1,000 genomes. That'll get rid of a chunk. And then if you want to create your own filter, you can set up an analysis of just your controls and do that list like I just did to create a list that will always be accessible in other analyses. The reasons why you would want to do that rather than including those controls every single time is that when you start including 100, 200 controls in every analysis you do, it does slow things down. It'll slow down your analysis time. It'll slow down your filter changing time. It does a lot of that stuff. So, it's not that we don't have high performance machines, but you will see the degradation. It makes a big difference if you have 100 versus 200 samples. All right. So we talked about how to get set up. We talked about how to work with artifacts. Now let's talk about how to make the best use of filtering within the application. One of my mantras, along with artifacts are your enemy, the other thing you want to keep in mind is that you want to keep things simple. Okay, don't ask too many questions at once. Limit the analysis to filter settings that will probe for a specific hypothesis. If you're interested in just coding to start, then set up your filters to just look at coding. And then if you think, well, I want to go look in the non-coding space, go look in the non-coding space. Or if you want to go look at structural variance, go look at structural variance. But I think it's helpful to look at these all in isolation. If you try to look at everything at once, it's easy to get buried. Okay, we are working with big data here, and it's not hard to find yourself overwhelmed in just so much information. You don't know how to handle it. The same with genotypes. If you're not sure if it's recessive or if you're looking for a simple recessive or compound heterozygote recessive case or dominant, it's good to test these each individually. It's going to help. So one of the filter settings I'm going to mention here is the predicted deleterious filter. This is kind of the non-coding, coding, how much you want to include. This I'm just showing you is my preference. It's not the default in the application for what I like to include. We have essentially a clinical or kind of a somewhat of a clinical assessment up here at the top. So how pathogenic does something look? We recently re-implemented or re-engineered our ACMG guidelines here to make these calls. Here in the middle section, we have gain of function. I include pretty much everything here except the last two, copy number gain and microRNA binding site. Particularly this one's a little bit dubious because they're based on predictions. So that's just my own, conservative way of coming at the data. And then for the loss of function, it's really around coding and splice variants. To start, okay, so I'm just asking a very simple question: which variants are primarily coding? I mean, that's really what I'm asking here. If you want to just get specific around coding, you clear out all the boxes and you just do these bottom two down here in the loss of function section. Okay. Then you can come back, and if you want to do promoters, just do the promoter box. If you want to do structural variants, just do the structural variant box. Okay. Ask simple questions. It'll be easier for you to not get buried. The genetic analysis filter is another filter that extremely powerful, extremely flexible, but just keep in mind how it's working and make sure you don't overfilter. So this is kind of more of a a tip than a trick. I see a lot of people overfiltering out in the field, and you need to be careful about that. So anytime you do have cases and controls, you have to keep in mind that variants are being compared in this filter, not genotypes. So if there's any variant that meets the checked criteria over here, usually that's not where the problem is. The problem is that when people start to think about, oh, and I'll also check all these genotypes over here, you have to keep in mind we are going to remove any variant that participates in any of these genotypes, because this is set up usually by default as a keep and exclude. So what I'm trying to say is that, for example, we're going to keep homozygotes, we're going to keep haploinsufficient, hemizygotes, nullizygotes, etc., and then we're also going to then exclude those variants if they participate in any of these following genotypes over here. Okay, the way this is set up right now is fine if I'm looking for de novo mutations in my cases. But if I were interested, as I point out over here, if I'm interested in a change in zygosity, if I'm interested in something that's going from heterozygote in my control to homozygote, let's say I'm looking at a paired tumor normal, I'm going to filter it out, okay? Because we're going to keep the homozygote, but you know what? We're going to throw that variant away because it also exists as a heterozygote. So it'll be discarded and removed from the analysis the way this is set up right now. So you have to kind of think, how do I want to set this up? What I think the best thing to do is you just go, and it's a little tedious, I realize, but it's for cases, ask yourself, for each genotype, am I interested in any variant that participates in this genotype? So am I interested in any variant that's homozygous, any variant that's haploinsufficient, any variant that's hemizygous, etc. in my cases? And then the more careful place you need to be more careful is on the controls. Do I then want to discard any variant that's also heterozygote or also occurs that's haploinsufficient or hemizygous? And so this is where it's helpful, because then I would get to here and I'd say, do I want to discard heterozygotes in my controls? And I would say, no, not if I'm interested in a change in zygosity. So I would know that my filter settings are not correct, and I would then go back and adjust them accordingly. Okay, so don't just blindly go with the defaults here, because our defaults are kind of guessing at what you want to do. We have some defaults up here. You can click the set button to get there, and they will approximate the optimal settings. But it's better just to kind of walk through it, particularly on the control side, and just make sure you want to remove everything that's over here, because it gets grouped and then removed. All right. Just a couple other tips. I don't encourage people to use the compound heterozygote filter in the control side. So if I come back here, I rarely ever recommend, oops, kind of crossed out that compound heterozygote checkbox over here. And the reason for that is what I diagrammed down here below. Everything here in the red brackets is considered a compound heterozygote. And the brackets show you what those compound heterozygotes would look like. Okay, now if I filter out compound hets in my controls, I'm going to remove every single row that's displayed here, because every single row is a compound heterozygote in my controls. However, if I was interested in this variant right here because it occurs at a heterozygote state, and then let's say this is a parent trial, let's say this is a trio, I don't want to throw this out. This might be very interesting. So typically I don't recommend filtering on compound hets. I also don't usually exclude haploinsufficiency. Haploinsufficiency is basically when a heterozygote is known to cause a phenotype. This is a prediction that's inferred by evidence. So again, we've gone out to the literature, we've looked at mouse information, and when a heterozygote in mouse causes a phenotype in mouse, we infer that it's haploinsufficient. You know, that's great, but it doesn't always translate to humans. So, you know, humans may have that same heterozygote and be asymptomatic. So you don't necessarily want to go out and remove predicted haploinsufficiency. If I refer back to my drawing up here on the top, you know, this could be predicted to be haploinsufficient as well. But, you know, in this case here, I could have two controls that just are asymptomatic, but my case is has a disease. So I wouldn't want to filter this entire row out because we deem that these are heterozygote haploinsufficient. So again, it's an opportunity to overfilter. I usually don't do that or the het. Also, excluding at the gene level, I'm going to go back a slide. You have an option to toggle this to variant or gene. I don't recommend doing gene for the same reason that the compound hets can be a problem. You can end up with kind of other sporadic variants. Well, now you have variants that are present in a gene. The whole gene gets thrown out, and now you just lost your data because you overfiltered. So those are just a few additional tips on using that genetic analysis filter. It's a great filter. You just need to be a little bit careful in walking through it. I'm not going to walk through this slide in detail, but if you want to do compound hets for trios, this is a scenario that I've worked out. So here I'm putting, again, I'm getting creative with the filters. I'm putting two genetic analysis filters together. One to keep heterozygotes in my child here. If I have two parents, I'm going to exclude genes that occur in both parents, either homozygous or het, because those would not be contributing to a compound het situation. And then down below, I actually switch my control side to keep, and then I'm going to keep hets that occur in both parents. But at this point, I filtered out when they occur in both parents at the same time. So now I'm going to filter for and keep places where those heterozygotes happen in both parents at different places in the gene. And thus we can end up with a compound heterozygote scenario. So that's just one example how you can really use this genetic filter in a very flexible way to get exactly what you want. So if I come over here, I've opened up a new analysis. I have this is a tumor normal comparison in my genetic analysis filter here. I'm set up for de novo. So you can see here in my results, I don't see any variance. I have this paired up. So I'm pairing tumor and normal, so they had the same subject ID when I set up the analysis. But if I wanted, I have options here. I can unpair, meaning that I can specify frequency in controls versus tumor, so it becomes more of a generic tumor versus control setup. And if I wanted to look for changes in zygosity, for example, as in my slides, we're going to want to change things. So if I want to see, gee, do I have any predisposing heterozygous mutations that then become homozygous in my tumors? Well, if I'm going to do that, I'm going to want to change this up. I'm going to want to say, well, let's keep only heterozygotes, and let's just keep the question simple. And I want to see if if I also see homozygotes. I guess hemi or noli would qualify over in my cases, and apply this. And you can see now I can see some genes where that might be interesting in the context of a predisposing mutation. Maybe not, but I'm looking for that. I'm asking the question. I think the P53 one is interesting because it does go to a hemizygous state here in my tumor. So this is my normal. This is my tumor. So just an example how to kind of walk through that genetic analysis filter. So keep it simple. Don't try to analyze everything at once. Particularly with the genetic analysis filter, avoid overfiltering. Don't just blindly use the default settings or any other settings. Think about what you're doing. And obviously, when you're doing the paired tumor normal samples, make sure they have the same subject ID, or you won't get that pairing. You won't have that option. Okay. Last little bit here, a couple more topics I want to talk about. Assessing variance. So, what variants are most compelling? Well, we talked about non-compelling ones if they're in the highly variable regions of the genome. And again, you can put your filters at the bottom of the cascade to kind of filter those in and out and see which ones are passing the confidence filter. Another one is display some of our tools, SIFT, polyphen, philo-P. They're all in the application here, so I can come over here, go to my Edit Columns button. SIFT is there by default, but I can add polyphen 2 in my philo-P conservation value. I can save these as default if I want. I don't, because I like to show people how you can do it. And now I have those three results over here to assess. So I can look at this P53 now and say, oh, is this damaging? It's damaging by SIFT, benign by polyphen 2. It has a so-so conservation score. But in some ways, it has a little bit more information than these other ones that are both predicted to be tolerated and benign. Be aware, both SIFT and polyphen have a pretty, well, I mean, they're good, but they're not perfect, right? So having those in my analysis, I typically don't filter for those. I just display those columns in my analysis. Okay. Do any of these variants have literature findings? Has it been seen before? Has it been reported in the literature? Here's a P53 finding. If I go to my variant finding column, I can see this has 26 variant findings in it. I can click on the variant findings link, and here I can dig right in, and I can see how much evidence is there that this is an interesting and biologically compelling variant. The more data there is, the more compelling it becomes. Okay. As we saw before, though, you need to be careful because sometimes even artifacts will have, been reported in the literature as being potentially significant. So you have to make that judgment call. Another thing you can do is assess the evidence in the path to phenotype view. So this is typically if I'm using my biological context filter. So let's pretend I'm going to expand this out a little bit. Hopefully you guys are all familiar with the biological context filter. I have a pathway that I'm using as my molecular, my biological context here. If I want to take a look at genes that interact with genes in my cancer pathway, I can do that, and I can click the Apply button here. So I get a lot more variants, and I have 81 variants. But then some of these are not going to be particularly interesting if I think about it in terms of a biological evidence. So, for example, if I look at this like UBR4 here, I can go to the path to phenotype, and I can see how UBR4 interacts with genes that are known to be part of my molecular mechanism of cancer. So I'm going one hop up from my pathway now. These are my pathway genes. Here's a regulator of those pathway genes. But if I click on these lines, it's binding. This is a binding of BRC2, which is UBR4. Or sorry, BRC is here. Sorry, BRC2 is here, UBR4. It's a binding relationship. Each one of these is a binding relationship. They're okay, but they're not as strong in some ways, just from a biological context pattern, or way of thinking as if they were biologically relevant. And one way you can increase the relevancy is, again, we're using our knowledge base here and asking, well, gee, you know, are there any variants that are upstream of my cancer pathway that actually cause or inhibit a loss of function? So now I am looking at a causal relationship, and it makes that biological relationship even stronger because it's not just a reporting finding, but it's a binding interaction, but it's an actual cause and effect. So now if I click on, let's say, HES3, and I go to path to phenotype here, and now I look at the relationship between HES3 and Wnt1. Okay, now I see homozygous mutant HES1 knockout in mouse, homozygous mutant HES3 knockout decreases expression of this Wnt1 mRNA and some cell type here. Okay, so this is now I can see, well, HES3 along with HES1 interact with Wnt and affect its expression. So the fact that it affects its expression, I know that's been shown, makes it a more biologically compelling relationship. So that's another way I kind of build an assessment of what looks to be biologically compelling. It's by, you know, assessing the evidence in that path to phenotype. Another way I go is I check and see how many variants are in my gene. If I go back, I'm going to take off my hop here. You can see that I'm going over on time a little bit. I'll try to wrap up here real quick. Let's see, I didn't do that right. I want to go effect. Another little trick I do is I go look up, and I'm kind of going back to, well, what looks noisy and what doesn't look noisy. If I just come up here to common variants, so this technically right here, these are uncommon variants. I'm going to click up here in my filter cascade. And then I can type in a gene name. I'm going to use quotes here so it does an exact match. So I can do SOS2. Oops, forgot my second quote. Let's do the second quote here. All right, so I can see I have 50 SOS2s in my analysis that are unlikely, as in they're not common. You know, that's okay. That's kind of high, actually. If I compare that with something, let's say my TP53 variant, again, I forgot my quotes. The quotes will do an exact gene lookup. You can see my TP53 mutation is much more aware of only 14 variants across the gene. And I realize that, you know, different genes are different sizes and whatnot, but this is just a quick back of the envelope way of assessing kind of how noisy that gene appears to be within my particular analysis. So here we're going to go look at frizzled 7. You see frizzled 7 only has 3 variants. So P53 and frizzled 7 look much more interesting in comparison to SOS2. SOS2 looks more artifactual. That being said, this frizzled doesn't look particularly interesting from an effect point of view, but could be combining with something else. So looking to see how many variants you have in a gene, key. Another thing I'll point out is just the ability to look at BAM alignment. So if I wanted to look at, you know, this, I could then click this IGV. You need to have IGV installed. IGV is an integrated genomics viewer from the Broad Institute. You will need to have access to your indexed BAM files. You do not upload those. Please do not try. It's a whole bunch of data we don't want. But you may have it sitting on a hard drive or a server that's local to you. You can upload that data into IGV, and by using that IGV link, go look at your analysis. And this is a screenshot I took from that viewer. And you know, it really helps, I think, to just go look at it. For example, so here we have a kind of the chromosome view up top. We have kind of a more contiggy view here in the middle here, and then you can see down here all the reads that span a particular variant. So here we have a heterozygote being displayed, and it looks real clean. It goes across lots of different sequences. Okay, as opposed to, you know, you see something like this that doesn't look quite so compelling. It looks like artifact. In fact, all this stuff over here looks pretty dirty to me. It's all happening at the end of sequences. It just doesn't have that same clean appearance and non-randomness across these sequences. So having a look at these can save you a lot of time before you go off and resequence. So again, that IGV link is right here, so that can also help in your variant assessment. So assessment considerations, does it come out in that variable in the confidence filter? If you do the variable, highly variable region filter, take a look at the SIFT polyphen 2 and phyllo-P assessments. Is there any literature around it? That certainly helps. What's the evidence when you do a path to phenotype? Okay, how compelling does that look? Check to see how many variants are in the gene as a whole by going, and these are going to be infrequent. So go to the common variant filter and then check that out, and go look at the alignments. I would do all of this before I spend a nickel on additional sequencing. The last thing that I often get is how to save. Went through that kind of fast. So how to save versions. I told you, ask simple questions. Well, what do you want to do when you want to save those results? Or if I have filter settings because I want to use those same filter settings over and over. So let me show you how you can do that. Oops, popped away a little too fast. So one way to save multiple versions is to share the current working with yourself. I'm going to come back here to my file here. So let's say I love my filter cascade. But you know what? I want to go off now and I want to change my genetic filter back to that de novo setting. Okay? But before I do that, I want to save my results here where I'm looking at these 5 variants. Hopefully everybody knows you can use the share button to share with others, so I can share my analysis and share my samples with other people, and when I do that I'm actually sharing a copy, but I can also share it with me. I trust myself, usually. So I'm going to type in my name there and share. So I typed everything correctly. It's usually the first step you must do. So now I just shared my analysis with me. And I would also be getting an email right now if I had my email turned on. If everything works out well, I can come back to see my analyses. I'm going to hit the refresh. Hopefully this will work out. It's not working out for me today. Usually I can hit the refresh and then it'll ask me to see if it'll, no, okay. Let me just try one more time here. Oh, here we go. So this is asking me, do I want to import that analysis I shared with myself? Yes, I do. So I'm going to import that now. And close this. And now I have it up here. So now I can come in here and I can edit and I can say, hey, you know what? This is my breast cancer, let's just call it, chains in zygosity analysis. I can save that. Okay, so that's now, and basically I've saved off that analysis. This is still open. I can just click right back here. Okay, so I've saved this version, and now I can come back into my genetic analysis filter and make tweaks. Okay, and then when I just close this, because the saving is done real time, I will then have my de novo filter basically as my working version. So that's an easy way to kind of save off versions. I told you to do that. I recommend doing that and save those versions off and then annotate them while you're thinking about it. The other thing people will ask me is, well, how do I save my filter settings? Gee, it would sure be great if I could click a save button up here in my filter cascade. We don't allow you to do that quite yet, but here's what you can do. When you set up a new analysis, so let's just say I saved an analysis, which I just did, but let's just say instead of calling this my zygosity change analysis, I just call this my template. Okay, I renamed this breast cancer analysis template. Now if I had new breast cancer samples coming into my analysis, I can hit, see here, let me go back to my samples. I'm just going to grab, here, I'll grab my random set here. I can click my Analyze button and put my name in, or my, and now I'm going to use this button. So instead of saying genetic cancer, etc., I can say use settings from analysis, and I could grab that one I just saved, okay, and make this my template that I use every time from here forward as my filter template, and then click next, and away I go and set up my analysis. Okay, so that way I'm using the filter settings from another analysis to then reuse them. So you don't do it from inside the analysis. You save the entire analysis, and I would just label it template filters. And then when I set up the new analysis, use those settings. So as I indicated, adjust your filter settings as desired, share the current working version with yourself. View, I go back, I hit the view analyses, hit that refresh, you'll get that import window, rename it, and then specify when you set up your new analysis. All right, so boy, I took almost an hour. I didn't realize it was going to take that long to go through all that, but hopefully a bunch of that stuff was helpful for you. We talked about how to get started, how to set things up quickly, how to manage samples. I know sometimes that can be a bear when you don't have folders to stick things in, but annotation is key. We talked about how to deal with artifacts. That's a biggie. How to use the filters effectively, particularly the genetic analysis filter and predicted deleterious filter. You saw me throughout be creative, I hope, kind of use things in different ways, stick things at the end sometimes. We talked about how to assess variance and kind of build up a profile, like which ones look most important, and then of course how to save versions and filter settings. So thank you very much for all attending today. I worked hard to get this set together. There's a lot more that I could tell you about, but clearly we took most of the hour. Maybe if there's a couple minutes, we can. Maybe if Antonio has like one question, we can take it.
Okay, thank you very much, Daryl. We actually have a few questions. May take a bit more than a few minutes, but I guess what we can do is we can start with, I guess, the question from Mila. I can go ahead and I can unmute Mila. Hold on a second here. Mila had a question regarding tumor normal case control analysis. Where are we here? Okay, so Mila, are you there? Mila, are you there? You can speak into your mic. Okay, well, she's online, but she's not speaking to her mic. But anyway, her question was, when you do a tumor normal case control analysis, why only generate a list of variants in the tumors and not the controls?
I'm not exactly sure I understand the question, but let me just kind of give the long-winded answer. So it's kind of up to you what variants you want to look at. So a lot of times people are interested in doing essentially somatic subtraction. So you say, well, what's unique in my tumor that was not in my control sample? And normally when I do a tumor normal analysis, I will usually have them paired. So I know I took the pairing off of here. I don't necessarily recommend that. I just kind of did that for display. But normally I would leave them paired, and then I would say, well, show me what's in my tumors and not in my controls. But if your scientific question is like I had done here to ask, well, what variants were in my controls? As we saw that predisposing P53 mutation back here, which was in my controls, then again, it's just a tweaking of your genetic analysis filter to ask that question. So it's really up to you what you're interested in looking at and analyzing, and there should be enough flexibility in that filter to do either one. I would usually do them separate. Again, just me being me, I like to keep it simple. I would say, well, what's unique to my tumor? What came in, what was our, what genetic burden did my samples already have before they even became tumorigenic? So hopefully that answered that question.
Okay. Our next question is from Thomas, and I can go ahead and I can unmute Thomas here. Hold on a second. And Thomas's question was, how do you add a filter to require call quality in both samples, cases and controls, instead of or?
I didn't quite hear that because there was some noise on my line. Can you repeat that?
Sure. Thomas's question was, how do you add a filter to require call quality in both samples, cases, and controls instead of or?
So you're referring to up here, call quality is at least here or here, and you're saying it needs to be and. I think that is um, more or less inferred here. I mean, I would think, and, to be honest, I'm not 100% sure. I would expect the behavior to be. I know that it says or, but I would think if I set this up to be um, set them both up to 50, that they would both be filtering at 50. But I haven't actually done the test, so I'm not 100% sure I can answer that. I'd actually have to look into that and get back to you. I apologize. I've not actually separate them. If it is a true or, and if one is high, so long as one is high, that's okay. Um, there's probably the reason for doing that is because you want to be able to assess it in your controls. Maybe they didn't sequence as well, or there was some contamination, tumor normal contamination. So, but to be honest, I don't know. I would think one way of doing that actually just occurred to me would be um, if you put this filter twice and you specified 50 here, um, and then ran and then added the filter again and then put 50 on the other side, whenever things occur sequentially in the filter, they're going to be ANDed together. So in that case, you would end up with 50 in each. But to be honest, without working it out, I can't say for 100% certainty. But that would be my guess. If it's not ANDing it already, I know it says or there. Then I'd run two filters and see two subsequent filters in a row and see if that doesn't fix the problem.
Okay, since we're running a little bit over on time here, we'll just we'll handle one more question here. One more question here from Paul. He asks, how do you know the variant is an artifact?
You never know, and that's kind of the challenge. Like when I was looking at my PGP data, obviously if it's occurring, or at least maybe not obvious, but my assumption is if it's occurring at 50% in my random control sample set, um, it's probably it's probably artifact. It hasn't been reported in the other large databases, so it's probably something unique to my platform. That's an assumption I'm making. Life is full of assumptions, and so yeah, you don't really know. Hopefully, what I've done is kind of outline, particularly like in the variant assessment section as well, just the things that look more like artifact, the ones that I would say they kind of carry a yellow or red flag with them. It says these are more likely to be artifact than some of these others, and there's no black and white. This is definitely artifact or not until you go maybe resequence, Sanger sequence it or something. But what I've tried to do is just kind of give you some tips for some ways to minimize the artifacts so that what you're left looking at are the ones that have more confidence associated with them, and hopefully the biology that you're also looking at is in that set. Like I said, sequencing is not necessarily the cleanest and easiest data to work with, particularly in certain regions of the genome. We just have to, I think, admit the fact that certain areas just do not sequence well. Okay.
Okay, great. Thank you very much, Daryl. Again, I wanted to thank everyone for attending today. Again, this webinar will be recorded and will be available on our website. We'll also do a follow-up email, so if you do have additional questions, when you do receive that email, feel free to respond to that email with your questions. And again, it was a pleasure moderating today. Thank you very much, Daryl, for your presentation. I'm sure it was very, very valuable to all of our attendees. And again,
Thank you.
we thank you, and we'll see you next time.